In this video, we're going to talk about aromatic heterocycles. You can see that we have 4 examples of heterocycles here. And in each of these heterocycles, you'll notice that the tricky part is how we talk about the electron pairs, or how we count them towards Huckel's rule. So first, let's define what a heterocycle is. A heterocycle is any ring that has a non-carbon atom in it. So, for example, pyridine and pyrrole have nitrogens, a non-carbon atom, and then we also have an oxygen or a sulfur that can also be considered to be a heteroatom or a non-carbon atom. And since it's cyclic, we call it a heterocycle. Okay, so let's go through and look and see how we count electrons. So remember, we're looking for odd numbers of electrons pairs to determine if these are aromatic. So for pyridine, we have 1, 2, 3 pairs. Notice that we are not counting that lone pair on the nitrogen. Pyrrole, we do count the lone pair on the nitrogen. 1, 2, 3 lone pairs. And then for Furan, we also have 1, 2, 3 lone electron pairs that we count. Notice that we're not counting the other lone pair on the oxygen. And at Thiophene, we count the same way as Furan. 1, 2, 3. We only count one of the lone pairs on the heteroatom. Okay, so what's going on here? And I tried to sum up the basics in these 2 bullet points down here. So notice that when we have an heteroatom with a double bond, we're not counting it towards Huckel's rule. So I'm talking about pyridine. So the nitrogen is part of the double bond, so we're not counting the lone pair. Also notice that what ends up happening is if we have 2 lone pairs on a heteroatom, we're only counting 1 towards Huckel's rule. So that's what's happening in Furan and Thiophene. So what the heck is going on, and why are we doing it this way? So let's look at the hybridization and how that's affecting things. So first, let's look at pyridine. In pyridine, the nitrogen is sp2 hybridized. And you can see here that we're going to have 3 sp2 hybrid orbitals. So one sp2 hybrid orbital is going to come over here and form a bond to carbon. And then the other one is going to come here and form a bond to another carbon. And you can see, I can draw in the little loopies at the end so you can see what's going on. In orange, this is the other sp2 hybrid orbital, and that's where the lone pairs are. And if you're looking at this or that's what the lone pair is. If you're looking at this picture too, you can see that the p orbital up here contains one of the electrons from the pi system. So we've got 1, 2, 3 pairs of electrons. Those are the 6 electrons. So this is part of the double bond. Remember, the p orbital is going to be part of that pi bond. So all of the 6 electrons are in this ring system around the top. The lone pair is sticking out to the side in an sp2 hybrid orbital. So it's not involved in the pi ring system at all. It's sticking out towards the side. The p orbital is the one that's going to contain the electrons that are part of the aromatic pi system. Okay, so this seems like it makes sense for pyridine. And if we go back, you can see that pyridine clearly is going to be sp2 hybridized. But Pyrrole, Furan, and Thiophene are a little odd because if you're looking at them, and if we use our rules of hybridization that we learned back in 2300, you're looking at these and you're going, wait, aren't those sp3 hybridized? Right? But it's kind of interesting because this is a part of an aromatic system. This is going to be one of those exceptions to the rules that we learned back in 2300. So if we go and look at Pyrrole, the nitrogen in Pyrrole is actually going to be sp2 hybridized. So again, we have an sp2 hybrid orbital. In this case, it's bonded to the hydrogen, and then we have sp2 hybrid orbitals that are connected to each of the carbons. There's a p orbital, and the p orbital here is where the electrons are. So it's kind of one of those weird situations where we're getting that nitrogen to be sp2 hybridized, even though it looks like it should be sp3. And that's just because it becomes sp2 hybridized because it's going to help it form this aromatic ring system. So now that lone pair is going to be hanging out in a p orbital and contributing to the aromatic system. Let's move on to Furan. And Thiophene was equivalent to Furan you're just going to swap out the O for an S. And the same thing that's happening is the oxygen is, or in the case of thiophene, the sulfur, is going to be sp2 hybridized. When it's sp2 hybridized, we are going to have one p orbital that contains the electrons, and then we're going to have the other lone pair hanging out in sp2 hybrid. And the other sp2 hybrid orbitals are forming the bonds to the carbons. So because of the fact that we can get that aromatic system, that's why the oxygen in furan and thiophene are going to become sp2 hybridized. And this will now put one of those lone pairs in that p orbital, and so the p orbital, the electrons in the p orbital can share over the aromatic system, and now we have 6 electrons, or 3 pairs, in that system. Now, we can also have parts of a molecule that are aromatic, so the whole molecule doesn't have to be aromatic. And this is showing Lipitor, which is known for its ability to help reduce cholesterol. And you can see that it does actually have an aromatic system, an aromatic heterocycle in the ring. It also has a couple other benzene rings, and those are other parts of the molecule that are aromatic, but we don't have the whole system aromatic. Another common aromatic heterocycle that you're going to see is in DNA. So remember, DNA is going to have 4 bases that can make up that portion of the DNA. The base, of course, would be connected to a sugar, right? And then the sugar would be connected to a phosphate group, and that would be connected to the larger backbone. Each of the base pairs in DNA is going to, or each of the bases is going to pair with another base. And each of these is also going to be aromatic. You can see the counts above here. So you can see 10 pi electrons in G, 10 pi electrons in A, 6 in C, and 6 in T. And they're highlighted in red here. Now what's interesting about this is you can see again, we've got the lone pairs on the nitrogens that are contributing to the pi system. So if you think about what's going on, again, we have to have this nitrogen over here is not going to be sp3 hybridized. It's going to take on a different hybridization and become sp2 hybridized so that these electrons can be in a p orbital and contribute to the pi ring system. So if you want to draw a resonance contributor, we could draw arrows where we show that a major contributor is actually going to have a negative charge and then oxygen, right? So we're going to have a huge delta minus on the oxygen, and that's going to actually help with hydrogen bonding. So if we skip to the next slide, you can see the base pairs that are formed, and we can see that there's a very strong hydrogen bond between an oxygen and a hydrogen, and that's very strong partially because of that large resonance contributor. And you can also see here, this is showing kind of the pictures about how C and T match up and A and T match up. And you can see G and C are held together with 3 hydrogen bonds. A and T are held together with 2. And that hydrogen bonding interaction is very strong, so you have lots of hydrogen bonds throughout the base pairs of the DNA double helix. And it just really kind of shows the importance and the strength of the hydrogen bond.